My name is Jason Rodriguez.
And my name is Randa, and today we'll be presenting the determination of fatty acid concentrations in algae. Algae have several different potential uses. Some are in agricultural fields, in space, in supplements, and in biofuels. It's reasonable to assume when we study algae that different strains of algae contain different types of fatty acids. Our objective is then to determine the types and amounts of these fatty acids contained within algae from the class Eustigmatophyceae. This class is found native in Arkansas and in several other parts of the world. You can see these are different pictures of algae, and within them you can see the lipid bodies. This is what we base our phylogenetic classification off of. So to begin the analyzation and extraction of these, we have to grow our algae sample. Our algae is cultured in a growth chamber, and then these cultures are grown in modified Wood's agar. Fatty acids are extracted from the algae then through a series of steps, starting with freeze drying and filtration and extraction. Then we prepare the fatty acid methyl esters so that they can be run through a GCMS. Fatty acids are then statistically analyzed to determine the relative quantities of the fatty acids found in each strain.
We calculated the average fatty acid concentrations of the small number of samples we have, and we have a few figures. Here we have UP3 and R2. UP3 is found in the Upper Peninsula, and then we have R2, which comes from Europe, maybe. We could expect to see some dissimilarities in them, but there are a lot of concentrations that are relatively close to each other. For example, we have the C18s in these two, and then we have Chicot, which is actually from Lake Chicot, and we have Mary, which is from Minnesota. Again, you can see that there are some close concentrations, but there are also some that are very different. But in all four, the C25, which is eicosanoic acid, is very high. And this is important because it's used in a lot of medications, such as medication to lower triglycerides, and it's also a biochemical precursor to many hormones. The analysis isn't complete, and we expect to get more samples, but in order for us to get a more accurate statistical analysis, we need those samples before we can make any conclusions. We would like to thank the High Performance Structural Mass Spectrometry Laboratory at the University of Arkansas at Fayetteville. We'd like to thank the doctors Follies for their collection and the classification of the algae samples we use. And also, we would like to thank the Arkansas Space Grant Consortium for the funding.